Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, in a previous class, we have completed the topic that is the type of mutation of this chapter, that is the principle of inheritance and variation. Now, in this lecture, we will be talking about the different type of mutagens. So let's understand what are the mutagens and what different type of mutagens are there and what effects they cause actually. So if you talk about mutagen, they are nothing but a certain agents, okay, that are responsible for causing a mutation. So if we define it, if we define it, that is mutagen, then mutagen are the certain agent which cause or which produce artificially produce certain mutations. So if these are the certain agents which are responsible for causing or producing the mutation, then they are known as mutagens. They are also known as mutagenic agents. Okay. Now, these mutagens are of two types. This mutagen are of two types. Number one, that is physical mutagen. Number two, that is chemical mutagen. Let's discuss this two type of mutagen in detail. In physical mutation, actually, radiations are involved, and in chemical mutagens, chemical compounds, several chemical compounds are involved, which are responsible for causing this mutation. So now we are going to discuss first the physical mutagen. When we talk about physical mutagens, in this, if we talk, then we can say that radiation are the most important physical mutagen. They are the most important physical mutagen, that are the radiations. In this, Muller was the scientist whose name is H. Muller, who was the scientist who used X-ray for the first time for the first time to increase the rate of to increase the rate of mutation in Drosophila. Okay. So in Drosophila, first time to increase the rate of mutation, S.J. Muller were used this X-ray, and due to this use of this X-ray to increase the mutation rate, they opened an entirely new field, an entirely new field in inducing mutation, in inducing mutations, and that field is known as actinobiology. So H.J. Muller is Muller is considered father of actinobiology. Okay. Muller is considered as a father of actinobiology. Okay. Now, this natural radiation, if we say, which are coming from the cosmic rays of the sun, that are actually a main force of spontaneous mutation. But there are some rays which particularly cause this or induce this mutation. Okay. So if we talk in detail about this in more, then in a spectrum, the shorter wavelength have higher energy. In spectrum of wavelength, if we talk shorter wavelength, if we talk has higher energy. And if shorter wavelength had higher energy than the visible light, than the visible light. So those shorter wavelength which are lower than the shorter than the visible light, or we can say they are lower than the 400 to 700 nanometer wavelength of light, they have a high energy. In this shorter wavelength, then the visible light are divided into this shorter wavelength. If we talk, they are subdivided into two groups. They are divided into two groups. One is ionizing radiation, and another is the non-ionizing radiation. Okay, this two type of radiation, this shorter wavelength are divided. Actually, these all radiation they are in a small amount in environment, and they are known to cause actually some of the background radiations. They are background mutations. So when we see the effect of this ionizing radiation and non-ionizing radiation, we can see this ionizing radiation cause ionization, and this ionizing radiation include X-ray, gamma ray, alpha ray, and beta rays. Whereas non-ionizing radiation, if we talk, This include are the particular wavelength which have a longer wavelength but carry a lower energy, and the non-ionizing radiation include the UV rays. So if we see the effects of this both ionizing and non-ionizing radiation, let's talk about the effects. So first we are going to talk about the effects of ionizing radiation. When we talk about the effects of ionizing radiation, this, as we have studied, this include the X-ray, gamma ray, alpha ray, and beta rays. This ionizing radiation cause break in chromosome. They cause break in chromosome, and due to the break in chromosome, the cells having this mutation show abnormal cell division. Show abnormal cell division. If this mutation, or if these cells include gametes, they may be of abnormal, they may be abnormal or even die, or even die prematurely. And this ionizing radiation, if we talk, they can cause different type of cancer, different type of cancer, and the frequency of mutation means how tough this mutation is, or how much we can say dose of this mutation is, or how much effective this mutation or harmful is. That frequency of this mutation or induced mutation is known as directly proportional to doses of radiations. Okay, so these are the effects of ionizing radiation. Now, after this, we are going to talk about the effects of non-ionizing radiation. Effects of non-ionizing radiation, and we have studied just now that this non-ionizing radiation include UV rays. So, this are the somewhat longer wavelength. This non-ionizing radiation have longer wavelength than the ionizing radiation, but carry lower energy. But carry lower energy. And this lower energy is insufficient to induce ionization. Insufficient to induce ionization. Okay. Therefore, this non-ionizing radiation, such as UV rays, it is known that UV rays do not penetrate beyond human skin. And it is known that thymine dimer, that is pyrimidine, thymine is pyrimidine, but thymine dimer formation is one of the mutagenic effect major mutagenic effect of UV rays. And this disturb, because in DNA thymine can be single, it cannot be a monomer, it cannot be in a dimer form. But if this dimer forms due to the UV rays, this disturb DNA double helix, and thus it also disturb DNA replication. So this is the radiation, which is the first mutagen, physical mutagen, which are going to cause this particular way, the particular different effects, mutagenic effects. But the second mutagen which we are going to discuss, that are chemical mutagen. And if we talk about chemical mutagen, then here large number of chemical mutagen are known. Okay. So these are the chemicals which are responsible for causing or inducing a mutation. And this chemical mutagen are known that they are more injurious than radiation, or more harmful than radiation, we can say. The first chemical mutagen, first chemical mutagen was mustard gas, was mustard gas by was mustard gas used by C. Orbeck et al. During World War II. During World War II. And this chemical mutagen are placed in two categories, means they are of two type. Number one, those which are mutagenic, those which are mutagenic to both mutagenic to both replicating non-replicating DNA. So those which are mutagenic to both replicated and non-replicating DNA are, if we take example, then they are nothing but we can say a nitrous acid. So this nitrous acid affect both replicating and non-replicating DNA. How, that we will discuss. And another category, another type of chemical mutagen are those which are mutagenic only to replicating DNA. Only to replicating DNA. Example, acridine dye and base analog. Okay. So these are the particular which affect replicating DNA only. So what are the effect of this different chemical mutagen like nitrous acid, acridine dye and base analog. So effects we are going to discuss. So first we are going to discuss nitrous acid. What are the effects of this nitrous acid? So when we talk about nitrous acid, just we have discussed it is mutagenic to both replicating and non-replicating DNA, and it act directly by oxidative deamination of by oxidative deamination of adenine, guanine and cytosine bases, which contain amino group. Okay, so this amino containing group are undergo oxidative deamination, means amino group is removed. So in this, adenine is deaminated to hypoxanthine. Okay, and this hypoxanthine, if you talk, it is complementary to cytosine. So actually what is given? Adenine pair with thymine. But if adenine get converted into hypoxanthine, it is not going to pair with the cytosine because it is complement. It is not going to pair with the thymine where earlier adenine was pairing. So the particular bond between hypoxanthine and the T which is present opposite to the A will not form. So it will cause a particular difficulty in a replication. Same way, it is known that guanine is deaminated to xanthine, and this xanthine pairs with cytosine. So here the major mutagenic effect is not going to be observed because guanine itself pair with the cytosine and its modified xanthine is also going to pair with the cytosine. Then the cytosine is converted to uracil and this uracil pairs with adenine. So actually initially it was cytosine which was pairing with the guanine, but when converted into the uracil, it is going to pair with adenine, not going to pair with the guanine. So bond will not establish and the DNA break will occur. So that is a nitrous acid. After nitrous acid, another chemical mutagen which show its chemical effects, that is acridines. And these acridines are very powerful mutagen. They intercalate or react between DNA bases and interfere with DNA replication. And interfere with DNA replication. So these acridines intercalate between DNA bases and interfere with the DNA replication and produce insertion or deletion or both of single bases respectively. So it interferes with the DNA, and in DNA either it will cause a insertion or deletion, or insertion deletion both respectively of a single basis individually. Okay, and if insertion deletion will occur, this induce friendship mutation or Gibbs mutation, and the example is thalassemia. Thalassemia is caused due to also this type of friendship mutations. Okay. Apart from this, the third mutating agent will affect the only replicating DNA that are base analog. And if we talk about base analogs, which are inducing a mutation and they are chemical mutagen, what are they? First, that we need to understand. So this base analog are actually somewhat structural similar forms of the normal bases. So they are what? They are the structural similar forms of normal bases, and they are incorporated in DNA only during DNA replication. Now what they do? They cause mispairing, and eventually give rise to mutations. This base analog can be either natural or artificial. can be either natural or artificial. Okay. If we talk about some of the natural base analog, then this natural base analog include 5-methyl cytosine, cytosine, 5-hydroxymethyl cytosine, and 6-methylpurine. Etc. can be the natural base analog. But there are some artificial base analog also. And if we talk about artificial base analog, that are 5 bromouracil and 2 aminopurine. These two are the artificial base analog which are most commonly used. So now if we talk about these two artificial base analog, you will come to know that this 5 bromouracil is base analog of thymine, means it gets incorporated in a newly replicated DNA in place of thymine. Okay. And 2-aminopurine, if we talk, it is artificial base analog of adenine. So it will act as a substitute of adenine and which can pair with the particularly cytosine as well. It is also observed. So if it is a base analog of adenine, it can pair with thymine. Why? Because it is a base analog of adenine, so adenine pairs with the thymine. But it can also pair with cytosine. That is due to the some structural defects or some structural changes we can say in structure of these 2-aminopurine such that it can pair with the cytosine as well. Okay, so these are the particularly we can say about the different base analog and different mutagens which are involved in the causing a mutation. Okay, so in previous class we have completed mutation, today we have completed mutagens. Okay, so this is the two important essential topics which we have covered. Okay, I hope you understood. Thank you.